So this is an artist's drawing of depicting the sexual life cycle, particularly in animals. In plants, it's a little bit different. It still has meiosis and it still has fertilization. So that's something that all sexual reproduction has. But we're going to look at the animal version of this as a good example, and probably one that you're somewhat familiar with in concept. So we want to make sure it's solid so that when you do learn about plants or fungus, you can see where the variation is. So we'll go through this one particularly. So something to think about, we have these terms, somatic cells and germline cells. A somatic cell is like a typical body cell, anything that's not part of the reproductive system. So they're not gametes, not eggs, not sperm. Somatic cells would be like skin cells, brain cells, blood cells, any of those types of cells, regular body cells. So most of your body is made up of somatic cells. And those kinds of cells, at least when you're young, they go through the cell cycle, and that makes more of them. And that's how you grow, and that's how cells are replaced when they're damaged. And so we talked about the purpose of the cell cycle for growth of the organism, because it makes more cells of the same type. But now we're looking at a different kind of cell division, meiosis, and that is restricted to just the cells that are what we call germline or reproductive cells. So in the animal body, most of the cells are somatic, and then a small group of cells in the testis or in the ovary can go through this other kind of cell division. And so meiosis, it's important, but it's not the most common kind of cell division in the body overall, but it is important because it's what produces the haploid gametes, which we call eggs or sperm. And so in this diagram, in the testis, there will be cells that go through meiosis, and that makes the sperm. In the ovary, there are cells that go through meiosis, and that makes the eggs. And then when an egg and sperm are meet, they fuse together, and that's called fertilization. I think y'all know that it doesn't shoot out of the top of the animal. It's happening in humans. It happens in the usually in the fallopian tube, and then it travels back down to the uterus, if I'm not incorrect. But in any case, it's internal fertilization. Some animals do external fertilization, like fish. But in any case, when those two cells fuse, the egg and the sperm, that makes a zygote, and the zygote grows through mitosis, through cell cycle, to form the new adult or the new individual. And so in this diagram, what sometimes throws students off, but I think if you think about it for a few seconds, you understand. When this individual here grows up, this baby does not grow up into one of its own parents. This baby grows up into another adult. So down here. Okay. So anyway, but it's considered a cycle because you keep producing a new adult or new individual each time. So it keeps going. Okay, so we're going to dig down into meiosis in terms of what are the events and what is the chronological order of the events. So the product of meiosis are these haploid cells, these eggs or sperm. And so what we're doing now is just digging into that process of meiosis and laying out what are the events and what's the order of the events. So meiosis actually has two divisions back to back. The first division is called meiosis 1, and the second division is called meiosis 2. Sometimes we use Roman numerals. In this slide, I didn't. But meiosis 1 and meiosis 2. And each of these phases has within it prophase, metaphase, anaphase, and telophase, and cytokinesis. But when we're in meiosis 1, we say prophase 1, metaphase 1, anaphase 1, telophase 1. And when we're in meiosis 2, there's prophase 2, metaphase 2, anaphase 2, and telophase 2. So when you see the numbers, that means it's a prophase or a metaphase or an anaphase or telophase of meiosis. Because when we use these subphase names for mitosis, we didn't have numbers. So we have kind of the same subphase names, but there's going to be some differences in what the events are compared to mitosis from chapter 10. So, don't sort of veg out here and think that you already know this, because there's some key differences here. And one of them is meiosis is not part of a cycle. Sexual reproduction is a cycle, but the cell division part is not. The cells, when they get to the end of meiosis 2, that's it for them. They can't go through meiosis again. So those cells, the products at the end of meiosis 2, the final product cells of all of meiosis, are the gametes, and they just kind of wait around and hope for fertilization. Okay, so meiosis 1 is called the reductional division. You saw that on your preview, on your vocabulary, but we're going to talk about what that means a little more. Meiosis 1 is called the reductional division. So the first meiotic division, meiosis 1, is called the reductional division. You're going to start with a cell typically that's diploid. That means it contains 2 full sets of chromosomes. So you remember what that means. 2 of each type of chromosome. And at the end of meiosis 1, you'll have 2 product cells, and each product cell will contain 1 full set of chromosomes. So the term haploid just means half of what you started with. So if haploid can be anything. If you start with a diploid cell, after one division, one reductional division, the 2 cells are each monoploid. So for diploid, a diploid starting cell, when that cell goes through a reductional division, the product cells are monoploid. And so in humans' case, that's the case. So for humans, haploid and monoploid mean the same thing, because half of 2 sets is 1 set. Haploid of diploid is monoploid. Okay, sorry. Okay, so now there's no DNA replication that occurs after meiosis 1 and before meiosis 2, because these divisions are back to back. And the second meiotic division is more similar to M phase of the cell cycle, mitosis. It's an equational division. So whatever you start with in an equational division, you end with 2 product cells that are also that same way. So in animals, you start with 2 haploid cells that end meiosis 1 will each go through meiosis 2 separately. And so each of those cells will divide again, but you start with 2 haploid cells and you end with 2 cells and 2 cells that are, all of them are each haploid. So you start at the beginning of meiosis 1 with any 1 haploid cell, you end with 2 haploid cells, and then over here another 1 haploid cell and 2 haploid cells. So you end up with 4 gametes from the entire process, and they're haploid. They each contain 1 set of chromosomes, and the chromosomes are in the monad form. So the products of meiosis are gametes, and then most gametes die. I wrote this down because I want students to think about this. In the world, most gametes do not get fertilized, and Professor Bittner is very glad about that. And that's true in any organism. Most gametes do not go on to that fertilization step. I mean, think about it. If every gamete that was produced by humans, eggs and sperm, got fertilized, then think about, the overpopulation of the earth. It's a good thing that gametes die because we just couldn't manage with that many individuals. So most gametes die. Meiosis happens, but then most gametes don't get fertilized. So let's go through the process of meiosis, and we're going to dig down a little bit into this meiosis 1 and meiosis 2 bit. But what happens in meiosis 1 is the separation of chromosome sets. So you start with a diploid cell, and at the end of meiosis 1, you have 2 haploid cells. But the diploid cell that you start with had dyad chromosomes, and the 2 haploid cells you end meiosis 1 with also have dyad chromosomes. And so what goes into meiosis 2 are cells that are haploid but have dyad chromosomes, and what you end at the end of meiosis 2 are cells that are haploid with monad chromosomes. So meiosis 1 divides the chromosome sets. Meiosis 2 divides the dyads into monads, I guess you could say, in the product cells. So watch for that to happen as we go through this. Okay, just like you know, prophase in the cell cycle is similar to prophase in meiosis. Prophase 1 has the same typical events of any prophase in that the chromosomes condense, they get tighter, they become more visible. The nuclear envelope disappears, disintegrates, goes away, and those spindle fibers start to develop. So those three things are the same three things that you learned about prophase in the cell cycle. But this is prophase 1 of meiosis, so we have a couple of other things that are going to happen. In prophase 1, the cell that starts meiosis always has dyad chromosomes. So the chromosomes are in the replicated form, starting meiosis. And each dyad consists of 2 sister chromatids. What happens in a diploid cell that starts prophase 1 with dyad form chromosomes is that the homologous chromosomes, the 2 dyads that are the same size and length, are going to come together. This never happens in mitosis. But in meiosis, the homologous chromosomes come together and we say they pair. But really what we mean is they touch. They actually lay on top of each other. I'll show you the next slide real quick. So this is a homologous pair of dyads, the blue dyad and the red dyad, and they actually touch so closely like this. And then what happens is called crossing over. So synapsis is just when they're touching or they're very close, and crossing over is the exchange of equal corresponding segments. And what that means is, I'll go to the next slide, a section, let's say this part of the blue chromosome breaks right here, and this same corresponding segment of the red copy breaks right here. So just on one sister chromatid of each of the homologous pair, they break in the same place and then they swap. So this red piece will end up attached to the rest of this blue chromosome, and the blue piece will end up attached to the rest of this red one. But there's really no problem there because remember, they have the same genes in the same order, so they're just swapping equal corresponding segments. So the synapsis is where they come together, and there's a lot of research on how they know, how they recognize each other. That's amazing. But then this breaking and swapping and rejoining is really amazing too. But this is called synapsis and crossing over. Now in metaphase 1, the homologous pairs still together are pushed to that midline, that metaphase plate. But what's different in metaphase 1 is how the spindle fibers are attached to the homologous pair. If you look here, you got to zoom in, but this one is attached to the dyad on the left, and this fiber is attached to the dyad on the right. So one fiber is attached to each of the dyads that make up the homologous pair. So one fiber attached to each. And then down here, one fiber attached to the red one fiber attached to the blue one. And you can see if you look real close, here's the result of that crossing over. They're trying to show that. But I want you to focus on how the fibers are attaching to the chromosomes. The homologous pairs are aligned at the metaphase plate in a side-by-side kind of arrangement. So those two are side by side, these two are side by side. Okay. So however many pairs you have. In humans, there'd be 23 pairs, 46 dyads, but 23 pairs. each pair has 2 dyads in it of the same kind, the same chromosome type. Now, the way that the chromosomes get pushed to the metaphase plate, in this diagram, they're suddenly showing 3 chromosome pairs, but in any case, we'll live with it. It can happen randomly. So when meiosis happens, let's say 8 separate times in the same organism, sometimes the chromosomes line up this way, sometimes they line up this way, or this way, or this way. So the way that the chromosomes line up is random. And what I mean is, like, they're colorizing the chromosomes, like the blue one and the red one of the same type. The long blue and the long red can line up such that the long blue is on the left and the long red is on the right, or it can go the other way. And that has no impact on how the short chromosomes line up. Is the blue on the left or the right for the short chromosomes? And that has no impact on this other pair that can line up with the blue on the left or the blue on the right. So all of these things are possible. Now, the 2 chromosomes of the same type are almost the same, but not identical. The homologous pair, they're not identical to each other. They have little tiny differences between them. And so what you're getting here are different combinations of long, medium, and short chromosomes that are lining up in slightly different ways. And this gives you that little bit of genetic variation as we move forward in the process. When you get to the final products, you'll be able to see some of what happens. So in anaphase, if you imagine the spindle fiber that's attached to each of the members of each homologous pair retract, and that pulls the homologous chromosomes apart. And so one set of chromosomes is pulled to one pole, and the other set is pulled towards the other pole. And so that's the separation of sets. So one of each type of chromosome moves to the left, one of each type of chromosome moves to the right. And by the end of meiosis 1, you have telophase, the reformation of the nuclear membrane, and you're going to have cytokinesis ultimately separating these 2 into 2 separate cells. Once cytokinesis has finished, you can see that this cell now has 1 long and 1 short chromosome, and this cell has 1 long and 1 short. So one of each type. And so each cell, each product cell at the end of telophase 1 is monoploid, or haploid in our case, same thing. But each cell still has dyad form chromosomes. And that's really what makes it possible for these cells now separately to go through another division. So meiosis 2 is much more like a mitotic division. Prophase 2 is going to involve the nuclear envelope dissolving and a new spindle forms. Metaphase 2 is going to involve the dyad chromosomes lining up singly, not in homologous pairs, singly, because the homologous chromosome is now in a different cell. So single file alignment of dyad chromosomes in any one cell. And then the sister chromatids in anaphase 2 are pulled apart. So that's very much like mitosis, where those 2 sister chromatids are ripped apart. And one of each type moves to the left or right. And by telophase 2, you end up with the reformation of the nuclear envelope. And so here they're following. These are the 2 cells that we ended meiosis 1 with, now go immediately into meiosis 2. So this is just a picture showing that. This is how the chromosomes line up in metaphase 2. And you can see now how the fibers are attached to each chromatid, which is different than meiosis 1. And so when these fibers retract, you get these chromosomes going this way, these ones going this way, and the same thing happening in this other cell. And by the end of telophase 2, once these cells are fully separated, you'll have 4 product cells, Each is monoploid, and each have monad form chromosomes. But what's true here is even though they have the same basic content, they don't have exactly the same versions of those chromosomes. This cell has a long chromosome that's mostly red with a bit of blue. This cell, though, has a chromosome that's completely just red and long. This one has mostly blue with a bit of red, and this one has just blue. So slightly different combinations here. So that's the genetic variation, these small differences on these chromosomes. These cells are not clones, and that's really critical. So the final result is that starting from one diploid cell at the beginning of meiosis, there are going to be 4 haploid product cells. In animals, these are going to develop directly into the gametes, the eggs and the sperm. In plants and in the fungi and in some of the protists, these are spores, but we'll let, when you go into other classes, you can worry about those things. But these are still going to eventually go to fertilization. So we're kind of focusing on the animal, definitely, not kind of, definitely focusing on the animal. So here you see a comparison of meiosis versus mitosis. The key things are, number one, the overall purpose. Meiosis is part of, but not the entirety of, sexual reproduction. But it is the cell division component of it, and it makes gametes. Mitosis is just involved with organism growth, cell replacement, the production of clones. And so the products and the purpose are very different. And you can see on the next slide, at the end of mitosis, you have 2 identical cells, still diploid. At the end of meiosis, you have 4 non-identical cells, haploid. So meiosis makes haploid cells that are non-identical. Mitosis makes diploid cells that are identical. So it's just very different, even though some of the phase names are similar. Okay, so to just emphasize some of the important points, the chromosomes behave, I guess you can call it behavior, is different in meiosis 1 compared to meiosis 2 and mitosis. So we really kind of focus on those events in meiosis 1 that are really different. And in meiosis 1, remember, the homologous chromosomes, synapse, they pair together, they come close together, and they cross over. That doesn't happen in meiosis 2, and it doesn't happen in mitosis. And then the sister chromatids are not pulled apart from each other in meiosis 1. They're not pulled apart until halfway through meiosis 2. And so that's important. So when you end meiosis 1, the chromosomes are still in the dyad form. And the product cells at the end of meiosis 1 have half as many chromosomes as the cell you started with, and so it's a haploid cell. So you have 2 haploid product cells at the end of meiosis 1, but they have dyad chromosomes. And by the end of meiosis 2, you have 4 haploid cells, but they have monad chromosomes. All right, the way that the spindle fibers attach to the chromosomes is different in meiosis 1 than it is in meiosis 2. I'll add that here, and in mitosis. So meiosis 2, and mitosis. Okay. So in meiosis 1, the way the fibers attach, one fiber, let's say from the left side of the cell, attaches to this dyad. The other fiber attaches to this dyad. Right? Same thing. And then in anaphase, they're pulled away from each other. But sister chromatids are not pulled apart. So we say homologous chromosomes are pulled apart. But what it means is that a whole set of chromosomes in the dyad form moves to the left pole, a whole set of dyad chromosomes moves towards the right pole. In meiosis II and in mitosis, the way the fibers attach, they attach to the sister chromatids of the dyad. So each dyad has 2 fibers attached to it. And when anaphase happens, they're pulled apart. Right, so that's specific for meiosis II and also for mitosis. So there's always this, like, compare what's happening in meiosis I, various things that are different compared to meiosis II and mitosis. So kind of to keep that in mind. So we said that sexual reproduction, remember that includes meiosis, which makes the gametes, and then fertilization, which brings those gametes back together in a fusion to make zygotes, increases genetic variation in the species. The meiosis has 2 mechanisms that generate increased genetic variation. One is called independent assortment, and one is the crossing over. So these 2 are both meiotic or cell division related aspects. And then we'll talk about the random fertilization last. Okay, so independent assortment is the idea that every time one of your cells goes through meiosis, your chromosome pairs can line up in any way, right or left, than any other chromosome pair. And what that ends up meaning is all those chromosomes are yours. So I'm a female, so my body makes eggs. So in the eggs, I can have, all of my eggs will have one set of chromosomes that are mine. But at the same time, my chromosomes, half of them came from my mom and half came from my dad. So they're all mine, but they're also in a sense, they're half my mom's and half my dad's. So the chromosomes that I pass on to my potential child in the egg there can be some eggs that have more of my dad's chromosomes than my mom's. And there are some eggs that could have more of my mom's chromosomes than my dad's, because there's just all things are possible. So it is possible for you to pass more of your mom's or dad's chromosomes to your child. And then a different child could get something completely different. So that's perfectly possible within the context of meiosis. Now, all of your children will get a set of chromosomes from you, but what extent they are also your mom's or your dad's, that's, variable. Then the crossing over is where, when your homologous pairs, one that was originally from your mom and one was from your dad, when they cross over, some of the chromosomes you pass to your children have part of your mom and part of your dad's chromosome spliced together. So that's even more interesting. So you have all these mechanisms that create these slight genetic differences in what's being passed to the next generation. And then random fertilization is the idea that any egg from one individual can fuse with any egg from another individual. Any egg from one individual can fuse with any sperm from another individual. And so because that fertilization can go in so many different ways, it's basically astronomical what kind of genetic combinations can be produced. So in the end, each zygote that is produced is genetically unique. In humans, they all have 46 chromosomes in the typical case, but within those 46 chromosomes are this combination of little differences. And so each zygote is genetically unique from that point of view.